be talking about this idea of genetic recombination. All right? And so, for us, how many different chromosomes do we have?
35? Something like that.
23. We have 23 pairs of chromosomes, right? All right, so humans have 23 pairs of chromosomes. Okay, and now we know that we each have one from our mom and our dad, and then when you go to produce offspring through meiosis, those get split, right? And so you can get any combination of those, all right? We have this law called the law of independent assortment. All right? And so basically all this means is it doesn't mean that so we have 23 chromosomes from our mom and 23 from our dad, right? We all understand that? It doesn't mean that when you split your gametes, okay, so when you make sperm cells or egg cells, okay, and they only have one pair, it doesn't mean all 23 of those are going to be the chromosomes you got from your dad, or all 23 of those are the ones you got from your mom, right? We can have any combination of these. So the law of independent assortment basically just means we can have any combination of chromosomes in a gamete. Okay? Now, can my phone do that? How many different possible gametes can we make if we have that many? So, we know that we have, you're going to end up getting two different sets, like either your mom's or your dad's, but there's 23 different pairs there, right? So let's do this. 2 to the 23rd power. Okay? So we've got 2 to the 23rd power. Anybody got a guess as to what this would be?
A lot.
Just take a guess.
1 million.
Oh, close.
2 million. Oh, you for sure real close.
8,003,000 or 300,000, 8,388,608. That's how many different types of gametes that one person can make. Okay? Gametes we can make. Okay? Due to genetic recombination. Okay? Now, what is this forgetting? We haven't done any crossing over here yet either. So this is, we can make this many different possible gametes without factoring in crossing over. All right? So, that means that the sperm cell or the egg cell that created you, there was a 1 in 8,388,608 chance that that one was the one that happened. Okay? Again, that's without factoring in crossing over. But then you have to factor in the other one, right? Because you get a sperm cell and an egg cell. So that means that the fact that you are the way that you are, if we take 800, multiply that by 2, no, except there would be, ooh, I think the pressure just changed in here because my ears just went. Anyway, but you can get any combination of those, right? So it's not just that you're getting this times 2. It actually works out mathematically that there is about a 1 in 70 trillion chance that the egg and the sperm that created you were the ones that found each other.
Really?
Yep. Pretty
So
wild.
basically what you're saying is I'm pretty important.
You're all beautiful miracles.
You're a special, not important.
Okay? So, but that's this whole process called, when we look at it, it's 2 to the n, where n equals pairs of chromosomes. All right? So is there any questions about that so far? No. Cool, perfect. That's what I like to hear. All right, and so we have these organisms. Now, we've got one organism that's going to have big A, big A, big B, little b, right? And then we'll have another organism that has little a, little a, little b, little b. Okay? Now, if we were going to do a Punnett square, okay, the only thing that this one can even give, right, is little a's and little b's, so we don't have to go with a full 4 by 4. We don't need to have a 16. Okay? We're just going to do a 1 by 4. Okay? Right? Because we know that the only thing that can give is A and B. Little a, little b. Right? Are we all good there? And so, bump this up. Okay, so if we go our 1, 2, A, B, we have 1A, 1B, 2A, 2B. Basically, this is just going to look like big A, big B, big A, little b, big A, big B, big A, little b. Right? We're all good there? Okay? And so, we would have in this box, big A, big B, little b, big A, big, little a, little b, big A, little a, big B, little b, big A, little a, little b, little b. Okay? And so we'd end up having two things here. Okay? You're either going to have normal wings and black color, or normal wings and white, or normal wings and black color, right? Because we don't have enough for the vestigial. There's always a big A in all of those. We can see that? Okay. And this will come out to be 50, 50, all right? And now remind you, he's doing this experiment, he's going to make thousands of fruit fly babies. The more data you have, the closer it should be to your predictions, right? Okay, so it should be 50% this, 50% that.
Again, understood the work of Mendel, and so he set up his Punnett square like this. And so on the top, he's going to show all the possible gametes that we could get from this one parent. So you could have both of the wild-type genes, or you could have both of those recessive mutant genes, or you could have a combination of the two. So you could have one wild, one recessive, or vice versa. Now this parent over here only can give its recessive genes, and so we could represent that on the other side like this. And so we knew that there are only 4 possibilities that we could get out of this. And those first 2 are going to look like that. And we call those what are called the parental phenotypes. Why is that? Because this one looks like that parent, and this one looks like that parent. In other words, there's no recombination. But on these other 2 alternatives, right here and right here, what we're getting is a recombination of those parents, and so we call these simply the recombinant phenotypes. But that shouldn't have been confusing to him. If we look at the Punnett square, we have 4 different squares, and so we would expect that 50% are going to be parental and 50% are going to be recombinants. But when he did this cross, what he found is that there were actually 17% recombinants and 83% that were of the parental type. And so was Mendel wrong? Was all of this wrong?
So he ended up finding he actually got 83% and 17%. Okay? And so why was this? When you have new organisms?
No, it's just that the model wasn't good enough. And so he thought about this idea of 17% and what it meant for a really, really long time. And then finally, one of his students, Alfred Sturtevant, and I couldn't find a good open source picture of him, but he's always smoking a pipe, so we'll say this represents Alfred Sturtevant, one night just blows off his homework and he figures it out. The whole thing he figures it out. And to understand it, you really have to understand what's going on during meiosis. And so if we look at these 2 parents, so this is the double mutant on this side, and then this is the hybrid on the other side. Let's look at each of those and figure out what gametes could they produce. And so if we look at the one on the right, we know that it can only produce these 2 gametes, but since we're seeing a frequency of recombination that's less than 50%, that implies that these 2 genes are found on the same chromosome. We know this now. Thomas Hunt Morgan and Alfred Sturtevant.
Okay, so that what he just said there is important. Can I erase this stuff right here?
Yes.
Okay. So, when recombination occurs at less than 50%, okay? And this is for a large sample size, all right? 2 genes must be on the same chromosome. Right? Because if it's 50%, we know that there's like, you have 1 from your mom, 1 from your dad, you can be getting those anywhere, right? They can, half of that's going to go one way, half of that's going to go the other way. But if we have something like this 17%, that means that they are more likely than not going to stay together, all right?
Had to kind of work through this. But if we look at what does that mean, these 2 genes are found on the same exact chromosome. So if we go through all the steps of meiosis, remember what happens first during interphase is that we copy all of the DNA, and then it divides in half, and then it divides in half again. And since those genes were on the same chromosome, I see just one possible gamete that could be produced. In other words, you're going to get one of each of those recessive genes. Now let's look at that hybrid pair. And we know, and Thomas Hunt Morgan knew, since he saw some of those recombinants, we had to have all 4 of these possible gametes. And so let's put the dominant or the wild type genes on this one chromosome and the recessive on another. So how do I know that I have both of the wild type on one chromosome and both of the recessive on another? Remember, this is the F1 generation, and so it's receiving this chromosome from a parent that was pure for both of these genes and vice versa for the pure mutant parent as well. And so let's go through the steps of meiosis again. And so what happens during interphase is that we copy them. Then there's one division, and then there's another division. And so how many gametes do you see? Well, this one is exactly the same as that one. And it's not based on orientation of the chromosomes because, again, they're both found on the same chromosome. And so this was puzzling, but then eventually they settled on this idea of crossing over. what if there were crossing over between these chromosomes? What if somehow this chromosome wrapped around this chromosome during meiosis, and they could see that under the microscope, they could see this occurring. If these crossed over, what you could get is bits of this chromosome actually being crossed over to that one. And so what we could now produce is a chromosome that has the wild type for coloration, but it has the recessive gene for this vestigial wing, and vice versa over here. And so Sturdivant, it's brilliant coming to this.
So all this is showing is that these two would have had to have crossed over to make these possible gametes that we would see in our organisms, okay? But what is the likelihood, okay, that this piece crosses over without this piece, all right? How close are they? And that's what we're going to look at here.
This kind of idea that if that crossing over event occurs between the different genes, then we would have recombination, genetic recombination. But if it doesn't occur during that, in that part of the chromosome, there's going to be no recombination. And so where does that 17% come from? Well, this is roughly 17% of that area of the chromosome. That's where it's coming from. If those genes were closer together, that frequency recombination would be closer. If they were really far apart, it's more likely that it's going to split in the middle. And so we can use this one cross to figure out the frequency of recombination, and then they were able to use that to build a gene map. And so if you look at a chromosome, if we look at that frequency of recombination.
Okay, so the 17% they found was basically saying that on a chromosome, these two genes are about 17% of the chromosome apart from each other. Okay, so basically that's just saying the higher this percentage, okay, if we have 17%, that means they're about 17, and we'll learn that they change percentages into map units, all right? Means that they're 17 map units away. If we have something that's 6%, okay, if there's a 6% chance that there's recombination, are those going to be closer or farther away?
Wouldn't they almost be closer?
They would be closer, right? Basically, this is saying there's a 17% chance that those genes are going to cross over without the other one because of how close they are to each other. If there's only a 6% chance that they cross over without each other, that means they're closer, right? Because you're moving part of that gene or part of that chromosome over. You're switching it when you do crossing over, okay?
Combination, let's say it's 17%, that implies that it's an arbitrary distance of 17 map units apart on the chromosome. Let's say the frequency of recombination is less than that. That means the genes are closer together. What if the frequency of recombination is greater than that? It means that it's farther apart. What if it's exactly 50%? Remember, that's what we were thinking about. If it was independent assortment, that would mean that those two genes are found on different chromosomes. And so we can use that to really map a chromosome. And so let's look at some of the data that they gathered. They found that the distance between the vestigial and that black coloration gene, the frequency of recombination is 17%. They then compared that to another gene called the cinnabar, which has to do with the eye coloration of the fruit fly, and they got these frequency of recombinations as well. And so when you're figuring out...
So, we're going to go ahead and make a chromosome map real fast, all right? We can use recombination percentages to make a gene map. Okay, and so all that a gene map is a map of the chromosome how far each gene is from each other. Okay? And so, we know that on our chromosome, we know that we have vestigial, okay, so we'll have VB or VG, and B are going to be 17 away from each other. 17 map units, right? Now we need to figure out where this cinnabar, the eye-colored gene, is. All right? So we know that it is what? We know it's 8% away from the vestigial trait, and we know that it is 9 away from the black trait. And so you want to start with one of them, though, okay? And what you're going to do is you would draw, let's go ahead and just use the vestigial one, all right? And we're going to put it 8% on either side, okay? And so if we have our cinnabar trait, okay, and let's just say it was 8 here, and we had our cinnabar trait, and it was 8 here, okay? So both of these are possible, but if we do 17 plus 8, what do we get? 24? No, 25. 25, right? And we know that the cinnabar is not 25 away from B. But if we take 17 minus 8, what do we get? 9, okay? So then you can get rid of this one. And you can show that this is roughly what the shape or the map of your chromosome would look like. All right? And so, that's basically what he's going to say here.
Figuring out a gene map, what I would encourage you to do is always start with the highest frequency of recombination. So I'm going to start with this one and just choose to put them on that chromosome, we'll say 17 units apart. So we're going to put the vestigial and the black apart by 17. Now let's go to another one. So let's figure out where these cinnabars are. Well, if we start with the vestigial gene, we know it's going to be 8 map units apart from that. So I could say maybe it's going to be over here, or I could say it's going to be over here. So we have these two different alternatives. And so which of those actually fits with that last frequency of recombination? Well, if I put it way over here, then we're going to have a frequency of recombination between that and the black. We know it to be 9%, but it's going to be a way larger number than 9%. And so I can narrow it down to this is where our gene map fits. Now let me give you a problem of your own. So now I've given you these 4 genes and their frequency of recombination. I would encourage you to pause the video here and then you try to
So on your guys' thing, I want you guys to try and make this, all right? I'll come back and we will do this together, but I want you to try and make your own gene map using those 4 numbers, okay? Start with the biggest one. Okay, so we always start with the biggest one, right? We have B and C is 45. Okay, so we're going to put those apart from each other. Now, we don't have A and D, so we can't do that last one yet, but let's go ahead and do A and B. Okay? Now we know that we can put A and B, we can put A 30 over here, right? Or we could put A 30 over here. Okay? Do we have anything between A and C? No. So we don't know that one for sure yet. All right? We have to use B and D now. Okay? Let's check B and D. Okay, so we'll put D will go this way, okay? Or we can have a D. Although D would be 40 this way.
Where's the, other part of the chromosome? Like, you got one line?
I'm just using a line as the chromosome. We don't need to make a bar every time.
Yeah, I know, but like, oh, I see, I got you.
I just using a line. All right, so these can be a couple of different places. All right, and this would be A over here. So now we need to look at the difference between A and D. Okay? So if we have A here and D here, what is that going to put it at? That would only be about 10 different right there, correct? Now let's just say we have 30 here, 40 there. I put it at 70. So AB is 30.
Well, you could have these.
45.
Charles, you could put it either way on that, as long as you put A and B both sides
Correct.
on the same side.
Oh, that's right.
So you could
Here,
put it
before
either
I
way.
confuse everybody, I'm just going to show his. It's
Map out
comes
where
from
each of those
C.
genes are found on the chromosome. I'll pause, and then let me show you what the right answer is. And so what I would do is, again, start with the largest frequency of recombination. I'm going to put B in.
That's what I did wrong.
I was gonna say.
Because you have to come from C on one of those.
B and C really far apart. So I'll put B on one side, C on the other. What's the total distance of the chromosome? Remember, it's going to be 50 map units. And now I could work backwards. And so now let me figure out. So I've got B and C. Where's D going to be? Well, I can't put D way out here because I don't have enough map units to do that. So I'm going to have to put it over here.
That's where I
And
made
once
a
I've got
mistake.
D,
I forgot that it was only 50 map units, possibly long. So let's expand this a little bit. Okay, so our chromosome is a total, remember, don't forget that, I forgot that, total of 50 map units, okay? And so we'll put B here and C here. Okay, so we know those are 45. Now, like he said, we're doing B and D, the 40. We know we can't go 40 this way, right? So, D is going to be like right here. We know the difference between those. It's going to be about 40. Okay. Sorry, I completely spaced that. It's only 50.
I got to figure out where A is. So I could work backwards to that. Well, I know that A can't be way out here on this side, so I know that A is going to be somewhere over here. So that would be the relative map distance, or the.
So,
Wouldn't the A be closer to the D though? He kind of, because it's only 25% away from the D and it's 30% away from the B.
so let's just go ahead and work that out real fast. We know that A and D are 25 away, right? So we put A is going to be like right around here. A is 25. Okay? And so, which one are we missing? We've got B and D are 40, A and D are 25, and B and C are 45. Which one haven't we used yet? A and B. Okay? And so, A and B, which doesn't work out perfectly, and these don't always work out perfectly, but that would be 20 there, right? And so,
The relative gene map based on frequency of recombination. And so Sturdivant and Morgan
that's
did
the
that over
relative
years, and they
gene
were
map.
able
It
to
doesn't
map
all,
out
the
where the
worksheets
genes
that
are
we're going to
found
work
in
out
chromosomes.
will
And
always
we
work
don't do
out
it this way
perfectly.
anymore. What do we do today? We simply sequence the DNA. Once we sequence the DNA, we can figure out where the genes are. But the cool thing is that as we compare that, you could go right here to the fly base. I was looking up the vestigial gene. We know exactly where it is, but that maps up perfectly with the work of Morgan and Sturtevant. And so that's genetic recombination. It allows us to create gene maps, and I hope that was helpful.
What we're going to be talking about here is, we kind of mentioned, or you guys have looked at it a little bit with between the videos or the documentaries and your paper, but I just really want to quickly talk about genetic disorders. Okay, genetic disorders are basically diseases or problems due to genetics. All right, and there are a couple of different types of these. All right, and so the first one we're going to talk about is going to be whether it is a recessive or dominant genetic disorder. Okay, and so these are going to be diseases you have, or issues you have. I guess I shouldn't always say disease, because it's not always a disease. It can just be a problem. You have if both of your alleles for a trait are recessive. Okay? So if you have two recessive things for, I think sickle cell anemia is one of them. But cystic fibrosis, there's a good one, where you build up fluid in your lungs, all right? And you need to get that bashed out basically every day. Or you could kind of drown in your own lung fluid. Yeah, not great. That is a recessive genetic disorder. You can get that if both of your parents pass down to you one of those traits, okay? Which, we talked about it, but the royal people over in England, right, and or just in Europe in general, the French, the Germans, the Russians, the English, all of them were pretty much all cousins, okay? And they would marry each other a lot to preserve the royal bloodline. And a lot of them had this thing called hemophilia. Basically, your blood doesn't clot. You could get a paper cut and bleed out, all right? A lot of the royals would often look sickly and frail because they had a lot of recessive genetic disorders. Now, let's say one of your parents has it, right? They have, or in order to have this recessive genetic disorder, this is what I'm trying to say, you need both of these things, right? You need two recessive traits. What happens if you just have one? Yeah, okay? So, if you have one dominant and one recessive trait, for this gene, okay? So you're heterozygous. You won't have the disease, okay? You don't have disease. But you are what is known as a carrier. Right? Because you could potentially, let's say, you and your husband both don't have, or you and your wife, Jordan was the one who said something, so that's what I was thinking. Your partner, okay, neither of you have cystic fibrosis, right? And so you think, oh, there's no problem here. You guys have a kid together, your kid comes out with cystic fibrosis. This would mean that both you and your partner had the trait. You were both carriers for it, okay? And you both pass down that recessive gene. And if you both pass down the recessive gene, they will then have this. All right, so carriers are a big issue when it comes to passing down genetic disorders because you don't really know if you have it unless you get your gene sequence until you have a kid. Okay? Now on the opposite side, you have dominant genetic disorders. Okay? So dominant genetic disorders, this basically just means if you have at least one dominant allele, you have the disease.
So Schultz,
Yeah.
if you and your partner both have the recessive allele, and then you have a kid, and then would your kid have a dominant allele then of that trait?
What?
Okay, so say you have a recessive.
Both? Like you're homozygous recessive, or you just have one? You're a carrier.
Just your carrier.
So you're both carriers?
Yeah. And
You-
then so then would your kid have a dominant?
You're both carriers, right?
Yeah.
There's a 25% chance your kid will never even get the possibility of having that recessive one, right?
Yeah.
There's a 50% chance they're a carrier, 25% chance they get the disease.
So say they get the disease,
It wouldn't be dominant.
would they?
There's no dominance in there.
Yeah, I know, but would the kid have a dominant? Like would the kid?
There's no dominant traits. There's no possibility of them having a dominant trait if they have the disease. Okay? Do you see what I'm saying?
Yeah.
Here, you can't pass it down. 50% chance your kid's a carrier, but they won't be affected. 25% chance there's no dominant in there, and you get the disease. Okay? So, in this, with dominant genetic disorders, there is no carriers. You either have it or you don't, right? Because you only need one. Okay? Huntington's disease, I don't know if anybody did that. Basically, your brain and body begin to kind of break down over time. It's not really a fun thing. Dominant genetic disorder. Okay? Now, one of the things with dominant genetic disorders, they're much more rare. Why would they be more rare than a recessive?
Because you don't really
You
get
know
carriers.
if you have it.
You know if you have it. You don't have carriers, okay? Here, this can get passed down through generations, and it just kind of sneaks through without people knowing about it. Back before we had really any good medicine, if you got Huntington's disease or if you had one of these dominant things, you probably didn't live long enough to even pass down your genetics, right? So it kind of worked itself out that we got rid of these dominant genetic disorders for the most part because we were removed. Got removed from society due to them not being able to live very long. Okay,
Did the same thing with that leprosy?
kind of.
What's
Yeah,
leprosy or whatever?
leprosy you get really bad like skin lesions, and I think things can begin to fall off if it goes long enough.
Like
It's-
you're just gone.
Yeah.
Like you basically get like holes in your body, and then
Don't they send
Body
them off
part.
to some island somewhere?
yeah, I know they put a bunch of the lepers in like a I don't know, it's in the Bible. Just read it. Stuff happens. Jesus went and cured a bunch of leprosy or something like that. I don't know. I never read it. I just know it happens. There are any questions? So these are the main two. You've got dominants and you've got recessives. Okay? Now we need to talk about autosomal, sex-linked, and X-linked traits. Okay? We've got plenty of time. so okay, so autosomal. All right, humans have 23 chromosomes, right? 23 pairs. The 23rd pair is your XY chromosome. It determines whether you're male or female. The other 22, so 1 through 22, are called autosomal chromosomes. Okay? And so basically, with autosomal genetic disorders, these can be either recessive or dominant, okay? But males and females have the same likelihood of getting genetic disorder. Okay, now why would we have a higher chance of one getting it for the sex-linked? Or the X-linked? Okay? Okay, so remember, right? 50-50 being a male, 50-50 being a female. All right, and you get 1X if you're a male, and you get a Y, you get 2 X's if you're a female. Now, these X-linked traits are more prevalent in males. Why is that? And they're generally recessive.
It's carried in the Y.
Nope. Because it's carried on the X. Okay? So let's just say we have a sex-linked disease. Okay? The mothers, females, can be carriers. Alright? Because females can be carriers. Let's say we have one of our X's here has the genetic disorder on it. Okay? We'll just put a D above this one. Alright, but the male doesn't have anything and the female only has one of the two. Okay? And so she is unknowingly passing on, potentially, this genetic disorder. Now, there's a 50% chance, or there's a 25% chance that you'll have a female who is not a carrier, right? You'll have a 50% chance that you have a male that doesn't have it. 50% chance, or I keep saying 50, 25% chance that there's a female carrier. But males only have one X chromosome, okay? So, if this guy gets it, he doesn't have another X chromosome to block it, right? It is either kind of similar to the dominant genetic disorders. You either have it or you don't. Okay? And so here, they only have one X chromosome. Normal X cannot block like in females. Either have it or don't. What's the only way that a female could get this disease?
If their other X chromosome didn't block it.
Yeah, so basically you could have a mother like this, where they are a carrier, right? And they would be unknowingly having it. But the father would have to have the disease, right? It's the only possible way. So then you have this and that, okay? Everybody see how that would work? So that's the only possible way that you could give a genetic disease and get it as a female if it's a sex-linked or an X-linked trait. Okay? These are basically the ways that we track a person's lineage. Okay, one of the ways. You can also use it to track, I know they do it a lot for like horses and dogs and like bulls, but you can look at something's pedigree. Okay, and this is basically a chart that tracks an organism's lineage. Okay, and usually when we're tracking its lineage, we will also track traits of interest. Okay, and so with a pedigree, they generally. Okay, let's go through what these would look like. All right, so we would have a square. Right? Squares generally represent males. Okay? Alright? Circles are females. Okay? Now, if we're looking at a specific trait, so say it was something like cystic fibrosis, okay? If you have a male that's colored in, this is a male who has trait. Okay, circle, going to be female with trait. Alright, now, if it's a recessive genetic disorder, how would you draw someone who is a carrier? Half. Okay, so either this or this would be a carrier. Okay? Now, we have, what is it, 2 more things? Alright, so a straight line in between. So if we have like this and this represents a marriage. Okay? If you were to draw a line coming down, this represents new generation. Okay? And if you have something like this, okay, what do you think that would be?
Brothers and sisters.
Siblings, correct. Alright? You can also use these to kind of backtrack and figure out what a person's genetics might be like. Alright? Let's say we have 2 seemingly normal parents, alright, in our pedigree, and they have four kids, alright? You're going to have a girl, yeah, let's go 2 girls, 2 boys. Easy enough. Alright, now, if our 2 girls and 2 boys, let's say out of this, We have a girl who gets it and a boy who gets it. All right? Now our parents didn't have it, but what does that probably mean about our parents? They're both carriers, right? Okay. Now, what do we know about these two then? We don't know anything. Because we don't know. Maybe they both have the two good traits, right? If they're both carriers, again,
So to know if you're a carrier, you have to have kids first.
or get your genome sequence,
get your-
basically. Okay? These guys, so right here, we know the genotypes of our parents, right? They're both heterozygous. We know the genotypes for these two. They're both recessive, homozygous recessive. For these guys, we don't know what they are. We do know one thing about them. We know they both have at least one big A and then an undetermined second thing. All right? These could both be big A's. They could both be little a's, and they could both be carriers. One of them could have two big A's. One of them could have a little a, and only one of them be a carrier, right? But we don't know that. So, when you're unsure about something, but you can tell that they have at least one dominant trait, you would write something like that. Okay? Do you guys see how this works?
Yeah.
Now, let's just say this guy has a kid with a lady, and they have one kid, and it's a girl, and this girl has it, right? Now we can fill in the blanks. This person was a carrier, this lady also a carrier, so then we put an A. Okay, but you have to kind of either, like Evan said, either get your genetics sequenced or have a kid. All right? But we don't know what's going on over here. Is everybody seeing how this works?
Yeah.
Okay, so